morning to all of you, and welcome to the second webinar of the Bracco Group, Beyond Life Science, Research and Innovation in the Era of Precision Medicine. We are here to take another step on the road that leads to Expo 2020 Dubai, the largest universal exhibition that is hosted by different countries every 5 years. In 2015, if you remember, it was in Milan. The Expo 2020 Dubai will open on October 1st, 21. It was due to open last year, but due to the pandemic, its inauguration has been postponed. During an expo, each country has the opportunity to showcase their best practice to the rest of the world. And the Italian pavilion has developed the theme Beauty Connects People. Let me say that we really need beauty now, and we really need to connect again with each other, especially after these months so difficult for everyone. The Italian pavilion has chosen Bracco as partner for scientific cluster. Together, they will celebrate the competencies in life science, the excellence in research and innovation, which have always been part of the Bracco Group's DNA. The Bracco Group is in fact a world leader in diagnostic imaging, medical devices, and advanced systems for contrast agents and drug delivery. This event represents one of the many activities that Bracco Group has decided to implement to raise awareness on the frontiers of medicine. Today, we will talk about precision medicine, which means taking into account individual variability in genes, environment, and lifestyles of patients. And of course, we will talk about the role of diagnostic imaging in this area. We all know, in fact, that imaging is a fundamental element of precision medicine that allows specialists to guarantee each patient the most suitable, tailor-made care. To address this issue, we have speakers of extraordinary competence from all over the world. But before introducing them, it is with great pleasure that I give the floor to Fulvio Renaldi Bracco, CEO of Bracco Imaging, who has been so kind to join us for a brief opening speech. Please, Mr. Renaldi Bracco.
Thank you for the introduction, and good morning, everyone. I warmly salute all the guests of this virtual meeting, which is the second appointment on the path to our Expo Dubai 2020, where Italy will have a central role thanks to the worldwide success of the 2015 Milan edition. I'm very proud that the Bracco Group is a gold sponsor of the Italian Pavilion, as we want to be proponents on the topics of future medicine and life sciences. Never has it been clearer than at this time in history that life sciences play a strategic role in people's health and are a key resource for ensuring a successful national economy. Therefore, I firmly believe that the right moment to act by focusing on innovation, especially in this area, is now. For me, that means ensuring broad access to the most innovative medical and diagnostic technologies for all patients that can benefit from it, as well as increasing a culture of prevention and developing new tools for early diagnosis as well as for therapy monitoring. This can be achieved through precision medicine, a new paradigm for the treatment of patients. More customized and individual approaches on single patients will highlight the specific characteristics of each individual in terms of genetics, lifestyle, as well as the environment. Precision medicine gives medical professionals the resources they need to target the specific treatment of the illness they encounter, further develop scientific and medical research, and keep families healthier. How this ambitious goal can be reached? By tailoring the right therapeutic strategy for the right person at the right time, identifying any predisposition to disease, and delivering timely and targeted prevention. So, if since about 2010 we have been talking about precision medicine as a distant futuristic breakthrough, it now represents the immediate future, the one around the corner. And given the context in which we now live, we can't afford to waste any more time. In this process of evolving technologies and patient needs, the role of diagnostic is crucial and represents the real link between the two parts, I would like to say that precision medicine depends on diagnostics. It is what allows precise data to be obtained for the formulation of a personalized and more accurate therapy. There can be no personalized therapy without precision diagnostics. In Bracco, we have deeply understood the evolution of the future of medicine, and for this reason, as a leading company in diagnostics, in the last 10 years, we have evaluated innovative solutions at the preclinical levels in early research phases. Most importantly, we have decided to invest more in this field. This reason why today we are carrying out important programs in the field of precision diagnostic with strategic investments for the development of new products in nuclear medicine, in ultrasound, and in fluorescent guided surgery. Now, before concluding, let me say that I am proud that we boldly dive into the future, seizing always new opportunities. Thank you all for your attention and hope to see you soon at the Italian Pavilion in Dubai.
Thank you, Mr. Renaldi Bracco. And now I would like to introduce to our guests Paola Testori Coggi, Special Advisor of National Technological Cluster on Life Sciences, Alisei, Cluster Alisei. Please, Ms. Testori.
Ladies and gentlemen, precision medicine represents one of the most innovative concepts in biomedical research and healthcare. It enables more effective early diagnosis, better and less toxic treatments, and improved medical services for patients. It can also serve as a potent engine to drive economic growth because it promotes innovation in diagnosis, therapy, prevention, and advanced technologies. And it ensures a more efficient use of resources while guaranteeing the best treatment for patients. Precision medicine is an area which benefits greatly from the knowledge transfer between basic research and clinical research, and where new diagnostic treatments and products can reach the patients thanks to the translational research. Precision medicine approaches are already being developed for diagnosis and treatment of a number of cancer and rare diseases, but their potential is still to be exploited fully. In fact, the success of precision medicine rests on some critical factors which I would like to point out. First, the development of personalized medicine relies heavily on integrated big data analytics and ICT solution in order to generate and integrate the required knowledge and infrastructure for new approaches. Second, the integration of all the omics data are necessary to generate and implement meaningful intervention and diagnosis. And these processes should be supported by reclassifying diseases at the molecular level and by developing new clinical trial design and new biomarkers. Third, innovative precision medicine solutions represents a higher uncertainty regarding going to the market. It is important, therefore, to apply new risk-based approaches for their evaluation and to encourage systematic early dialogue with all stakeholders in order to provide the best guidance for companies. Collaborative and transdisciplinary research, as well as cross-sector collaborations and multidisciplinary projects, will help to address all these challenges. Therefore, any occasion to share knowledge and to create new alliances and networks at worldwide level is crucial. To this aim, the Italian cluster on life sciences Alisei, that gather academia, research institute, and industrial association, will participate to the Italian Pavilion of Dubai 2020 and is looking forward to give its contribution to the development of precision medicines and its integration in the healthcare system. Thank you.
Thank you, Ms. Testori. It is now time to introduce you our scientific panelists in alphabetic order. So the speakers will be Valentina Giannini, Senior Researcher, Imaging Lab Department, Surgical Sciences, University of Turin. Fabian Kissling, Chair of Experimental Molecular Imaging at RWTH Aachen University in Germany. Ilan Misano, Telemedicine and MedTech Researcher, Israel Europe Technology Transfer Consultant. David Schuster, MD Nuclear Radiologist, Emory University School of Medicine of Atlanta, USA. And Fabio Tedoldi, Director of R&D of Bracco Imaging, Italy. And Mr. Tedoldi, I would like to start with you. This is my question. We heard loud and clear from Dr. Rinaldi that there can be no personalized therapy without personalized diagnosis. Can you help us to understand a little bit more the role played by diagnostics in the precision medicine arena, and what the contribution of the R&D of a pharma company specialized in imaging solutions like Bracco can be?
Sure. Thank you, Elisa, for your question, and good afternoon, everybody. It's my pleasure to be here today and to open this scientific session in preparation of the Expo event in Dubai. Yes, the statement from Dr. Rinaldi is very appropriate, I believe, because the concept of tailoring a certain therapy is well known. But what is changing today, actually, is the availability of data and information collected by several diagnostic techniques, which make our capacity of tailoring this treatment make our capacity much stronger. So, as an example, you can see here how much the cost for full sequences of a human genome has decreased from 20 years ago to nowadays. So something that was, let's say, available only for research purpose in the past or for very few people can be available almost for every one of us. But also closest to our daily life, probably all of us has experience in these wearable devices, which are intelligent devices that can collect a number of parameters from our body, from our physiology, including the heart rate or the blood pressure or the amount of calories which are burned in a day. And so this all this information from genes, from the environment, from our lifestyle, can be combined with also with additional information that comes from imaging techniques. So from those techniques that we are somehow used, like X-ray or magnetic resonance, or ultrasound, nuclear medicine techniques. And imaging plays his role in different part of the diagnostic arena. So from screening to early diagnosis, to guidance of treatment, to follow-up of therapies, and also to recognize possible recurrences of diseases. And today, I simply want to provide you with a couple of examples that can give you the feeling of how powerful nowadays in the era of molecular imaging all these techniques can be. And I will start with an example based on early diagnosis, but even more on the possibility of doing a follow-up of a therapy. And well, this is a picture from a very well-known paper, one of the most cited in the scientific literature, which is titled The Hallmark of Cancer. And one of our research projects in our Bracco R&D nowadays is focused on the so-called detection of the angiogenesis. So what is angiogenesis? Angiogenesis is the, let's say, growing of new vessel which are needed somehow in a tumor to feed tumor cells and to make them growing very fast. And there are a number of so-called anti-angiogenetic treatment that by inhibiting this effect doesn't allow the growing of the tumor mass. And so one important player in angiogenesis is the so-called vascular endothelial growing factor, which is released by tumor cells, and that has the rule somehow to trigger the formation of neovessel once the VEGF is able to bind to the so-called vascular endothelial growing factor receptor. So it's a tandem effort between these two molecular entities to build new vessels. And anti-angiogenetic treatment usually acts on one of these two entities and try to stop, to block this interaction and thus the formation of new vessels. And in order to monitor the efficacy of this treatment, one of our projects is aiming at designing a specific ultrasound contrast agent, which is ultrasound contrast agent normally are so-called microbubbles, so a phospholipid shell which contain a gas, and this gas-solid interface make them visible to ultrasound. But let's say we are decorating these phospholipid shells with a peptide, so a relatively small molecule, which is capable to identify the vascular endothelial growing factor receptor 2 and make it visible to ultrasound. And you can see here on the right part of this slide how this appears in an ultrasound examination. So on the top left, you have the standard ultrasound without injection of contrast agent, then the contrast agent is injected, and you see here appearing this mass, which is the tumor itself. But that's not the main aim of our project. So the main aim is not to visualize the tumor itself, which is something that we can do with other technique or other contrast agent for ultrasound, but it's more to look specifically at angiogenesis and in particular to follow up the therapies to inhibit angiogenesis. And here we have a proof of concept of how this works. And we have administered to an animal model two different drugs, one which is a standard, let's say, chemotherapeutic drug, and the other one which is an anti-angiogenetic drug. And you see here on the top graph on the page, the blue line refers to control, and you see that both drugs represented in red and green are able to decrease the size of the tumor, but on the bottom part of the picture, you see that let's say the intensity of the ultrasound signal after injection of this new microbubble, which is specifically targeted to VEGFR2, is the signal is decreased only for the green line that correspond to the treatment with the anti-angiogenetic drug. This demonstrates that with this specific product, we can follow not only the size of the tumor, but the fact that the vessels are growing or are not growing. And so we can follow up this specific treatment. Well, you see, there's a lot of information here, and let's say the best result will be obtained if we will be able to manage all this information coming from genes, from environment, from lifestyle, from imaging in a proper way. And well, already today and even more in the future, we will have artificial intelligence helping us in this job. But I will not enter in any more detail because I know that, let's say, the following speakers will go more deeply into this topic. And instead, I want to now provide you with a second example, which is on a different field and how imaging can be used and how specifically new classes of contrast agent can be used to guide treatment. And the treatment that I want to focus on today is surgery. Surgery is still the most used treatment for cancer. It's in general very effective, but sometimes when surgeon leaves some part of the margin of the tumor still in the patient, so if there is a not complete resection of the tumor, you can have recurrence in many type of tumor, and this is very annoying first for the patient, and it's also very costly for our healthcare system. So what we are doing in this day, a second project that we want to present is a project on the so-called image-guided surgery. So we are designing specific devices that called camera and specific drugs or contrast agent which allows surgeon to visualize better the tumor and the boundary on the tumor and to be more precise in the resection of the mass with a final benefit for the patient and for the healthcare system. And to illustrate this, I would ask the director to put, let's say, the first of my videos.
Based on our research, we are developing tracers targeting the F receptor for fluorescent image-guided surgery in order to assist surgeons in identifying colorectal cancer. Image-guided surgery uses tracers consisting of antibodies or peptides coupled with fluorescent dyes which bind to proteins like the F receptors. When the tracer is excited by near-infrared light, it emits fluorescent lights, which can be detected using dedicated camera systems. After excitation, a fluorescent signal is seen on the surgical video monitor, indicating the presence of tumor tissue. Using this technique, the boundaries between the tumor and healthy tissue are clear. The surgeon can then remove all of the tumor, resulting in improved patient outcomes.
So very good. I hope that with these two examples that come from the type of research that we are doing in our R&D centers in Bracco, I was able to give you, let's say, a sense of how the imaging can, and in particular novel contrast agent, can help in diagnosis as well as in guiding treatment and to follow up treatments.
Thank you, Mr. Rinaldi. But going beyond diagnostics, we know that sometimes molecular drugs are inhibited to do their job simply because they do not physically reach the target. Is there any way to facilitate the permeation of drugs into tissues, which are particularly tricky to penetrate, and to make this patient-specific?
Yes, and this gives me the chance to talk of a third project that we are carrying on in our R&D groups. And I have again another slide here. Okay, so as you said, sometimes drugs are not working simply because they are not able to cross biological barriers. And probably the stronger biological barrier is the so-called blood-brain barrier that preserves our brain from infiltration or of unwanted molecules. But when we have to deliver a drug to a certain part of the brain, we need to open this barrier. And again, using the same platform that I shown before for ultrasound, so these microbubbles, okay, we are able to use them as a sort of resonator that push somehow on the blood-brain barrier and helps and facilitate the delivery of drugs into the brain parenchyma. And again, I have another small video here to help everybody to understand how it works. So if we can put the video.
Established a non-invasive approach to briefly increase the permeability of the blood-brain barrier in a controlled, targeted, and reversible manner. This procedure requires the use of MRI-guided focused ultrasound and the intravenous administration of phospholipid microspheres, also known as microbubbles. The ultrasound waves are generated by a transducer array fitted around the patient's head. With guidance from MRI, the transducers can be targeted to sonicate precise brain regions of interest. The drug to be delivered to the brain is injected into the bloodstream with the microbubbles. Drug and microbubbles travel throughout the circulation, and as such, once the acoustic waves reach the brain region of interest, they activate the microbubbles within the blood vessels at this location. The ultrasound has a frequency of millions of waves per second. Here, they are being shown at a much lower speed for clarity. The ultrasound waves interact with the microbubbles, which absorb the acoustic energy, causing them to expand and contract within the capillaries. This phenomenon is called oscillation, and it results in the stretching and compression of the capillary walls. As the permeability of the blood-brain barrier increases, the therapeutic drug begins to exit the capillary and enter into brain tissue. The duration of the sonication cycle is very short.
Thank you, Mr. Tedoldi, for your interesting presentation. And now it's my pleasure to give the floor to Ms. Valentina Giannini. Welcome, Ms. Giannini. My question is, we heard in a previous talk that an accurate characterization of oncological disease is really fundamental for the personalization of medicine. From your point of view, how can we understand the evolution of a tumor thanks to imaging, and how can we predict a therapeutic response?
Okay, yes, thank you for the invitation and for the kind introduction and for the question. I have some slides to present my point of view. And yes, cancer is a complex and heterogeneous disease. And we understood that there is that the same cure doesn't work the same on different patients, and even more on different lesions on the same patient. And images can play a critical role here to help classifying patients in different subgroups who share identical disease characteristics and a similar treatment response and prognosis. For example, let's take a look at these images here. This is a CT scan for a single patient with 9 liver metastases originated from the same rectal cancer. And imaging here is really important because it allowed to detect all these 9 metastases, which is very important to evaluate the current state of patients at the moment of the diagnosis. But imaging can do more. For example, if we look at just 3 of these 9 metastases, here, next slide. Here, you can see these 3 metastases, and they are quite similar in shape and color. For example, they are all dark compared to the liver, which is brighter. However, these metastases are quite different. Indeed, after 3 months of therapy, 2 of them increased their dimension, while the third one decreased its dimension, meaning that the third one responded somehow to the treatment, to the same treatment. In general, this is a patient with a mixed response, in which 6 over 9 metastases decreased their dimension, while 3 metastases increased their dimension, which means that they did not respond to the therapy. And this is quite a problem, and we want to predict which ones will respond to therapy. So imaging can do more. There must be something different among these metastases, and there are some hidden data that needs to be extracted by converting digital medical images into meanable high-dimensional data through a process called radiomics. Radiomics allows us to extract some parameters, some features that can describe not only the color of a single pixel, but also the relationships that exist between one pixel and its neighborhood. And this information can be combined using artificial intelligence to generate some signatures that have the potential to uncover disease characteristics, especially those that fail to be appreciated by the naked eye. And the hypothesis here is that radiomics can distinguish lesions that are born to be bad from those that are born to be good. And in this way, this could be exploited to bring the personalization of treatment to lesion level instead of patient level. And we tried to understand this concept. We started with a CT scan before treatment, and we applied some artificial intelligence algorithm to automatically segment the lesions. From these segmented lesions, we extracted 77 features, including volume and first and second order parameter, which means intensity of pixel and also relationship between pixels. And we applied other artificial intelligence algorithms to distinguish between metastases that will respond to therapy, to treatment, from those that will not respond to therapy. And if we take the patient I showed you before, we were able to correctly classify 8 over 9 metastases, and in particular, the few that did not respond to the therapy. And this is extremely important to allow to conduct different strategy on those few metastases. But we also learned something more from our model. We learned that homogeneity is somehow related to the response. Indeed, among the whole features, difference variances and homogeneity were those most important for the model. And we learned something. We observed that the 2 metastases with homogeneous pattern did not respond to the therapy, while the third one showed a more heterogeneous pattern. Of course, these homogeneous and heterogeneous patterns are not measured qualitatively, but they are assessed quantitatively in radiomics. But this is extremely important. So we learned 2 important things here. We learned that the model was able to correctly identify most of patients with outlier lesions. That is, that they did not respond in a general condition where most lesions respond. And this is important because in this case, biopsy could have revealed a different genetic makeup, prompting the use of a different target molecule, or suggested the use of a local ablation of outlier metastases. But we also learned that homogeneity is somehow related to response to therapy. Therefore, if our observation is confirmed, the identification of a more heterogeneous pattern could be predictive of response to therapy. Of course, the road to use these models into the clinical practice is still difficult and full of pitfalls, but the stakes are high. Imaging can really contribute to target the most suitable treatment according to the characteristic of each patient or even each tumor inside the same patient. Thank you.
Thank you, Ms. Giannini. I have another question with you because we know that your working group developed a prototype to improve the diagnosis, the observation, and the treatment of prostate cancer. Would you please tell us something about this new system?
Yes, I have some slides also here. And our prototype, based on artificial intelligence applied on magnetic resonance imaging, MRI, is very important in the management of patients with prostate cancer, which represent 14% of men worldwide. MRI as the technique with the highest accuracy in detecting clinically significant prostate cancer, up to 92%. Also, MRI can allow to plan target biopsy, which means that can help to identify a greater number of clinically significant cancer and avoiding the useless identification of indolent tumor, avoiding the biopsy in 1/3 of men, and also to overtreat this patient with dramatic side effects. MRI, for these reasons, in 2020, both the European and the American neurological associations strongly suggested the use of MRI before the first round of biopsy. And this is absolutely great for patients, but it will produce a large increase of MR examinations worldwide in the next few years for staging, active surveillance, and also for screening. And this could be a problem for the national health system because multiparametric MRI is a time-consuming task, since the radiology has to look to up to 1,000 of images per exams, including morphological and functional information. Moreover, the diagnosis is strongly dependent on reader expertise. Indeed, we demonstrated that radiologists could miss up to 1/3 tumors. And this is not good if we want to take MRI in a screening level. Therefore, our system really works well in this setting because it's able to automatically collect all sequences from the PACS and to apply some artificial intelligence algorithm to produce a likelihood malignancy map in which suspicious areas are selected. These suspicious areas are further analyzed to produce an aggressiveness score. And this score is very important because it will produce a change of treatment based on this score. But I will come back later to this score. Also, all this information automatically computed are filled into a structured report that contains information about patient, prostate, and also lesions. And all the important information of lesions are automatically computed and filled, such as size, dimension, and aggressiveness. This will strongly reduce the reading and reporting time. We demonstrated in a clinical setting that our system was able to increase the sensitivity in detecting high aggressive prostate cancer of 10% without affecting specificity. We also demonstrated that the reading time was reduced by 1/4 and inter-reader agreement increased between readers. And this is very important to diagnosis. Moreover, the most important things here, I believe, is the aggressiveness score that is produced. In that, this score is a 3-level score, in which that lead to a different behavior in treatment. Indeed, if a patient is classified as level 1, not likely to be aggressive, or level 3, likely to be aggressive, the urologist will have a clear indication about the treatment. I mean, not to treat level 1 or directly treat without passing from the dramatic side effects of repeated core biopsies. And this score is able somehow to substitute in the future, of course, the biopsy to a something called virtual biopsy, in which the real biopsy is not performed in patient classified as level 1 or level 3. And this will strongly reduce the number of biopsies performed worldwide. So In general, our fully automatic CAD was validated on a multi-center data set and reached high accuracy in discriminating between low and high aggressive tumor. Such tool could drastically, as I said, reduce reporting time with very important benefits to the patients. And I like to say that no aggressive tumor are classified as level 1, which means that no aggressive tumor will be undertreated with our system. Of course, here the validation is still to be performed, a larger validation, but results are extremely encouraging to a better personalized medicine. Thank you for your questions.
Thank you, Valentina Giannini, for your great presentation. And it is now my pleasure to give the floor to Mr. Kissling. Welcome, Mr. Kissling, and this is my first question. One of the most interesting direction of precision medicine is nanomedicine. What are we talking about? It is already a reality in the clinical practice, and if yes, what are its applications?
First of all, good morning together, and thank you very much for inviting me to join this exciting session. It's really an honor. I will now be happy to answer your question, and I prepared some slides to illustrate it. Often the word nano is connected with either positive and or negative emotions. But in principle, we are just talking about pharmaceuticals that have a size between 1 nanometer and 999 nanometer. And if you consider the scales, and what you see here on this slide is my son when he was about 1 meter, and compare the size of a 100 nanometer particle in my son, then the relation is almost equal as if you would compare my sun to its location on the Earth. So that means that we are here still talking about very small dimensions and very small particles. So why should we use nanomedicines, and what is the advantage? First of all, we can improve the delivery of drugs via nano-sized drug delivery systems. And in this context, we can take advantage of passive and active targeting mechanisms. We can use the enhanced permeability and retention effect that is very prominent in many tumors. We can slow the uptake and release of drugs at the disease place, so we can generate some kind of depot effect, or we can become much more cell or tissue specific. Furthermore, we can implement trigger drug release, either, for example, by pH, by temperature, by light, or by physical stimuli like ultrasound. Another important point is that we can also encapsulate multiple drugs that would, in principle, if you would just inject them, they would all have different kinetics. You can implement them in one drug delivery system, and then you can release them in same concentrations at the same time, which is very important for many combination therapies in precision medicine, in particular if you're talking also about immunotherapies of cancer. And finally, you can also add, because of the high versatility of nanomedicines, diagnostic tags to them that make them trackable in the body. There are really many synthetic, biologic, and hybrid systems in evaluation, and they work via very different targeting mechanisms. And here you just see an example of the different nanomedicines that are really common in products. And here we talk about, for example, about liposomes. We take about polymer drug conjugates. We talk about micelles. We also talk about antibody drug conjugates. We should never forget that antibodies, which are our natural, most powerful immunological defense mechanisms, or let's say bullets in the body, these are also nano-sized. They have a size of about 12 nanometer. We're also talking about dendrimers. We also talk about solid nanoparticles like iron oxides, gold nanoparticles, or small fluorescent nanosystems like quantum dots, or even gas-filled vesicles. And if you now think about how these can accumulate and compare it to a free duct, you can say that a free duct easily crosses the different biological compartments. So that means it goes very rapidly everywhere in and everywhere out. It has a very huge distribution volume, not only at the place of disease, but also in the healthy body, and therefore it can generate negative side effects. A nanosystem can be much more selective to areas where the vessel permeability is higher, or where you have an impaired lymphatic or venous drainage, which causes to a retention of the nanosystems in the diseased area, and this is called the enhanced permeability and retention effect. Then you can add targeting moieties to the nanoparticles so that they bind to either, for example, tumor cells or the activated endothelial cells, and thus render them more specific, and you can increase by this process also retention and uptake. And finally, I already mentioned that you can also apply external stimuli if the nanoparticles are properly designed to release the drugs only at the place of disease. You may also be interested whether nanosystems are already in the clinics, and the answer is clearly yes. We have already more than 20 tumor target drug delivery systems in clinical use. And in principle, the aim of them is to improve efficacy and to reduce toxicity like you want it from every drug. And for that purpose, you try to protect the drug from the body so that it's not already active or cleaved in the blood, or you want to protect the body from the drug so that you don't have unwanted side effects. And we have more than 20 tumor-targeted drug delivery systems of nano size in clinical application at the moment. And these are also as versatile as the different systems that I introduced on the previous slide. So you have also here polymer protein conjugates with drugs, drug antibody conjugates. You have protein drug nanoparticles from denaturated proteins, for example. You have pegylated liposomes, and you even have first multi-drug loaded liposomes in clinical application. If you consider the development of the nanomedicine market, so we can already say that about 18% of the complete nano fields are already pharmaceuticals. So that means it is a significant amount that is already for medical applications. And if you here consider the money spent on nanosystems, you see that there's a clearly increasing trend over the last years. Here's just an illustration what you can expect from nano. This is the accumulation of doxorubicin in comparison to a nano-based drug delivery system, and you very nicely see that doxorubicin accumulates if you inject it in its free form in the tumor, but it also has, for example, a high accumulation in the heart where it can cause severe side effects. However, if you encapsulate it in nano drug carriers, in this context, polymers were used, you see that you can enhance tumor uptake, but that the offset accumulation is strongly decreased. So what can we now expect from nano, and which sizes should we use, and what benefit can we expect from being more specific by targeting nanoparticles to the disease site? And I would like to show this on one example that we made several years ago. Here we used 4-arm polyethylene glycol systems as drug carriers, so a polymeric drug delivery system, and we used those systems with 6 nanometer diameter and with 40 nanometer diameter. You should note that with 6 nanometers, it is still cleared by the kidney and has a short blood half-life. With 40 nanometers, it has about the size of an IgG antibody, meaning our natural immunological nanosystem. And in order to target them, we bound riboflavin to 3 arms, which targets riboflavin receptors. These are vitamin receptors that are often overexpressed in cancer tissues. And additionally, we added a fluorescence tag to make them visible in the body. If we now inject these nanosystems into tumor-bearing animals, you can clearly see the benefit from the larger systems that have the antibody size. Here are the small ones, here are the large ones, and you see that due to passive accumulation and due to taking benefit of the enhanced permeability and retention effect that I explained before, you have significantly more tumor accumulation. However, you also see that with respect to accumulation, the active targeting only contributes to improved accumulation of the small probes. And the reason is very simple, because the small probes rapidly diffuse in the tissue back and forth and are eliminated rapidly. You have here a big advantage of the increased retention that you generate by letting the systems bind to the tumor cells. However, in the antibody-sized systems, the passive accumulation is so dominant that active binding does not significantly contribute to overall accumulation. So you may not say, okay, then active targeting does not make sense, and did nature make something wrong with our antibodies? No, the answer is clearly no, because if you not only look for over accumulation, but for cellular uptake and internalization, then you see for both systems that you have a significant benefit from actively targeting towards the tumor cells. That means you have from active targeting mostly a benefit on improving retention internalization. And this makes also sense for antibodies because you want to transport a high amount of antibodies to the diseased tissue where they should just lay around you. So you generate some kind of depot effect where they then slowly diffuse towards the diseased cells, bind, and either repair or kill them. This sounds very promising, but unfortunately there are many challenges in the nanomedicine field as well. Not all patients show these enhanced permeability and retention effect. So you need somehow to pre-select the right patient cohort that really takes benefit from a nano formulation. You often have the case that metastases behave different than primary tumors. You need to consider that as I already mentioned, not only accumulation is important, but also access to the tumor cells. And outside the vessels, you have still a very strong stromal barrier, which often restricts diffusion of the systems through the tissue. So you also need to consider penetration. And of course, you also have to reflect whether it makes sense for your system to do active targeting. This is often beneficial, but it can also be negative. For example, if you reduce the blood half-life by your active targeting moiety, then you may also reduce the passive accumulation. And at the end, you may come out with an active targeted probe less efficient than with a passive one. So all these things are difficult to consider and need really careful refinement.
Thank you. Thank you, Mr. Kisling. But there is another issue that I would like you to discuss with us, and it is theranostics. That is, when image diagnosis and therapy come together. Can you show us some examples of image-guided drug delivery?
Yes, thank you. Thank you very much for this question. This really brings me to my next slide. And I just talked about the challenges for nanomedicines, and I said you need to refine the systems and to refine the use and to pre-identify patient populations that really respond. And for that purpose, it is highly beneficial if you combine diagnostics with therapeutics in the same system. And then we are talking already about theranostic nanomedicine. So that means a theranostic nanomedicine is in principle just a therapeutic nanosystem that additionally has a diagnostic function. That means that you can see it and that you can determine its accumulation and distribution in the body. And this you can use to do prognostication, for example, of therapy success. You can apply it to pre-select patients to certain therapy schemes. You can use the information to adjust dosing, and of course also to monitor treatment response. So that means that theranostic applications are a very important element of precision medicine in the nano field. Let me give you an example. So these are theranostic pi cells, so pi stacked polymeric micelles that are developed by Dr. Shi and Professor Lammers in my department. You see that we can produce these systems with a very defined size. And we can also load them with imaging tags, either using tags for PET, SPECT, nuclear imaging, or for fluorescence. My next example shows you a fluorescence example, because in animals that's cheaper, and you can do it with higher throughput. And you see here tumor-bearing mice. And if we now load these micelles with cytostatic drugs, for example, with paclitaxel, and add an imaging tag, and then after end of therapy, look for the accumulation of the probes versus tumor sizes, we clearly see that if we had low accumulation of the nanosystems, we had big tumor sizes in dark green. But if we have high accumulation of the nanoprobes, this bluish yellow signal, we had very small tumor sizes. That means, in other words, you can use this information to predict, if you would do it otherwise around, if you would first inject the nanosystems, look, do I have high or low signals? Then you already know will the tumor respond well or not, and then you can decide whether you apply these systems or not, or whether you change to another therapy scheme. This is my last example, and I would also like to introduce you triggered drug release opportunities. And what you see here is a movie. It's not from me, it's from a publication from Healthfield. And you see this is an ultrasound microbubble that is stimulated by ultrasound pulses, and you see it's starting to oscillate, and it's strongly oscillating. And if a bubble in the body, in the bloodstream, oscillates like that, it transmits physical power to the environment. And this physical power is strong enough to open biological barriers, for example, to make vessels more permeable or to untighten the stromal barrier. So our idea was, why not using these bubbles to open the blood-brain barrier? So the brain is very closed. Here we have the strongest vessel barrier, because here the endothelial cells are somehow fused. We call that tight junctions. And this is a very strong barrier for many drugs that cannot be properly transported into the brain, which restricts the therapy not only on brain tumors, but also on other diseases like neurodegenerative diseases like Alzheimer or Parkinson. So therefore we thought, let's use these microbubbles to open the blood-brain barrier. So we injected them, we applied ultrasound. We were very successful. We could open the blood-brain barrier in all mice, and all mice died because of brain edema. So we thought, okay, the principle works, but we did much too much. We need to control the mechanism. We need to have a theranostic really to see how much and to which extent do we open the blood-brain barrier. And for that purpose, we embedded iron oxide nanoparticles in the polymeric shell of our microbubbles. You see it in the middle image, it's a TEM image. And then the idea was that if we apply ultrasound, these bubbles burst, then the iron oxides are released, and they then penetrate into the brain tissue that has been permeabilized. And then we can use MRI with T2 star-weighted sequences to monitor the signal change, which then should correlate with the degree of blood-brain barrier opening. Here you see that it really works. This is the change of the MR signal. You see that if we only inject microbubbles or only apply ultrasound, nothing happens. But if we do both together, we have a change in MR parameters that are correlated with the insonation time. That means the longer we insonate, the more the MR signal changes. And we also correlated that with the accumulation of a model drug. And this example, we used a 70 kilodalton fit stick strain. And you see here that this dextran clearly accumulates outside the brain vessels, indicating that we were really able with that approach to enter really the brain tissue. Even more, we could show that by varying the ultrasound power and the intonation time, we could even regulate how deep our drugs penetrate into the brain tissue. Even that could be regulated. So I think this was my last example, and I would like to summarize. So nanomedicines are certainly no magic bullets, but they are highly valuable for many diagnostic, therapeutic, and theranostic applications. They can significantly improve drug delivery and reduce overall drug toxicity. They offer you versatile options for functionalization, for example, to improve cellular uptake and retention by active targeting, as natural antibodies do. A big advantage of nanomedicines is certainly the combination of different drugs within one system. So you have big opportunities for modern and innovative combination therapies. But certainly, you often need to pre-select the right patient population for your nanomedicine treatment. So that means you really need to go deep into precision medicine, and for that purpose, you often need theranostic strategies. So with that, I would like to end. I thank you very much for the questions and for listening to my talk, and look forward to the next part of the session.
Thank you, Mr. Kissling, for your speech. And now I would like to give the floor to Mr. David Schuster. And my first question is, your research group is focusing on precision medicine. And in particular, you're working on the Emory Winship clinical trial with the PET radiotracer fluciclovine. What are the results of this study?
Well, I think first it would be helpful to define what precision medicine is. So I will now consult with Dr. Wikipedia for that definition. Precision medicine is a medical model that separates people into different groups, and medical decisions and interventions tailored to the individual patient based on their predicted response or risk of disease. And that certainly sounds nice, but is it more than just a catchphrase? So let's give that real-world example. Science often starts with a problem. And the problem here is that after treatment for prostate cancer, the cancer comes back. It's called recurrence. And that occurs in approximately 1/3 of patients. A sensitive blood test called PSA, or prostate-specific antigen, signals there is a recurrence, but does not tell us where the cancer has returned. Yet knowing where the cancer is will affect treatment. Typical imaging tests like MRI or a CAT scan are not very sensitive to detect the cancer. If we're using radiotherapy to treat the recurrence, we're left flying blind. And standard radiation fields must then be used. And these are really one-size-fits-all radiation fields. And they're only successful in about half the patients. So we like to say, to treat it, you have to see it. With better tools, we may determine where the cancer is coming back. And then we asked ourselves, if we can see it, what happens if we treat with personalized fields rather than standard or one-size-all-fits fields, like we see here? So how are we going to see it? Well, one tool that we can use is a PET scanner. And a PET scanner is not this, but it is positron emission tomography, where we inject the patient with a small amount of radioactive trace substance, or radiotracer, and we image on a powerful camera. So what radiotracer are we going to inject? Well, cancer needs to feed. And the menu includes amino acids, which are the building blocks of protein. Fluciclovine is a radioactive amino acid that is FDA approved to detect recurrent prostate cancer with PET. And this is an example of imaging with fluciclovine, where we have a nodule that is positive for recurrence, biopsy proven. The Empire 1 trial, or the Emory Molecular Prostate Imaging for Radiotherapy Enhancement trial, was conceived by my colleague, Dr. Ashish Jani from Radiation Oncology, and myself. It is a gold standard trial, so it's a randomized prospective design. 162 patients with suspected prostate cancer recurrence after prostatectomy were enrolled. Then they were randomly assigned to either arm 1, which had the usual radiation therapy, not personalized, or arm 2, in which the patients had personalized radiation therapy based on that fluciclovine PET scan. And then we wanted to see how the patients did over time. And indeed, the fluciclovine, or personalized arm, does better. By 4 years after radiotherapy, 50% of patients in the standard approach failed, but only 25% of those in the fluciclovine approach who had personalized planning failed. In other words, 3/4 did well. This is one of the few studies to show improved outcomes by using a personalized therapy approach guided by imaging. And this was not possible before we had these advanced imaging tools. And hence, we are now doing the Empire 2 trial, which is about halfway done. It's a similar concept, but one arm uses the fluciclovine to plan radiotherapy for patients who have that recurrence or biochemical recurrence after prostatectomy. And the other arm, and these patients are randomly assigned, uses a different PET radiotracer called PSMA, or prostate-specific membrane antigen, and we will be looking how these patients do over time as well.
Thank you, David. I have another question for you. In what ways do you think that imaging will play a role in the future of precision medicine, and will this include artificial intelligence or machine learning?
Well, that's a great question. So first, let's talk about some terms. And these are some ways in which I think the future is headed. First, there is theranostics, and it's a combination of diagnosis and therapeutics, and some of our other speakers have discussed this. We use imaging and other diagnostic tests to predict who will respond or not to targeted radioactive or non-radioactive therapies. And it's said that theranostic paves a way for the holistic transition from trial and error medicine to personalized medicine. And here I am quoting the authors of this paper. One example is PRRT, or peptide receptor radionuclide therapy, where we use a radiotracer directed against a certain receptor in a tumor, which emits low-level diagnostic radiation to see if the tumor is detected. In other words, does it have those receptors? And if so, we administer a similar radiotracer directed against that same receptor with destructive radiation. And here is an example with the tumor and the PET scanner. Then we give the diagnostic radiotracer. It attaches to the receptor, and then it's detected by the PET scanner. And then if so, we also give that destructive radio tracer, which can destroy the tumor, as we see here. One way in which we use this now is with neuroendocrine tumor. This is a type of cancer which caused the death of Steve Jobs, for example. And these commonly have these somatostatin receptors on their surface. And PET scan with the radiotracer dotatate lights up if the tumor has these somatostatin receptors, then the patient can get that PRRT directed against the somatostatin receptors. But if the neuroendocrine tumor becomes very aggressive, the tumor loses the somatostatin receptors and starts consuming a lot of sugar, which can then be detected with a common PET scan using a different radiotracer, a radioactive sugar tracer called FDG. In that case, PRRT will likely fail. So let's look at our first patient who has, and this is a PET scan with dotatate at the level of the liver, and there is a lesion in the liver, a tumor, which is taking up dotatate. It's bright. The spleen is also shown, but that's normal. And there's no uptake with FDG. So this is positive on dotatate, negative on FDG, and the patient is a good candidate for PRRT. The second patient has uptake in liver tumors with FDG. They're using that sugar, but there's no uptake with the dotatate. Therefore, this patient is positive on FDG, negative on dotatate, and is a poor candidate for PRRT. Another concept is something called multiparametric, where we can use powerful imaging devices, such as, for example, the newly invented combination PET and MR scanner, which can look at many aspects of the cancer at the same time. And these are just some examples. It's not exhaustive. Anatomy, blood flow, oxygen, water, and other molecular content, metabolism, receptors, inflammation. And multiparametric may also include non-imaging aspects, such as genetics. But in many senses, we are still staring at the shadows on the wall of Plato's cave. We just have many more shadows now to bring together and to interpret. So how can we put them all together in a better reflection of reality? Well, one way is with artificial intelligence or machine learning. And these are computer systems that are able to learn and adapt without following explicit instructions. And they use algorithms and statistical models to draw inferences from patterns in the data. And AI may help us better interpret the shadows and tie these different aspects of the tumor into more targeted therapy with improved outcomes. AI here is not used as a black box, replacing humans as some have advocated, but as an augmentation to human intelligence. So this finally brings us to cost versus benefit. And the question is, when do we need to visit Savile Row for a bespoke, perfect, but expensive tailored suit versus using less expensive off-the-rack clothing that may be good enough? And AI may help with determining how much personalization is needed for any one situation, for any one patient at any one time. How good is good enough?
Thank you, Mr. Schuster. And now I'd like to give the floor to Mr. Misano. Welcome. So my first question is, Dr. Schuster talked about the role of artificial intelligence in diagnostics and in precision medicine. What are the applications of artificial intelligence in clinical practice and in the everyday life of patients so as to improve the customization of diagnosis?
Thank you so much, Elisa, for your question, and thank you to Bracco Group for the invitation to this webinar. I am Ilan Misano. I am a biomedical engineer from the Technion Institute of Technology in Israel, and I am part of the Italian Technion Association. We are an association that links the Technion University with Italian universities and with Italian research institutes, and we create a bridge in this way between the high tech in Israel and in Italy and in Europe. Especially, we also help companies to see the innovation here in Israel, in the startup nation. So thank you so much for the question. I have some slides to show you. So we are speaking about artificial intelligence in medicine. Let's give a little background about artificial intelligence. How artificial intelligence works? It works with data. So at the beginning, we have to train an algorithm with data in order for the algorithm in the future to do precise predictions. So as we see, for example, here in the slide, we see a lot of pictures of tomatoes, and then we teach the AI that these are tomatoes. Okay, in this way, the AI learns how to associate the shape of a tomato to the name tomato. In this way, in the future, when the AI receives a picture with something that resembles a tomato, it's able to say this is a tomato. And this happens in the same way in medicine. We need a lot of data, a lot of true data, a lot of ground truth data in order to teach the AI to be as precise as possible. So the applications of AI in medicine are basically infinite. But I'm gonna present to you right now some of the most common applications of AI in medicine. But again, with the increase in computing power, for example, also with quantum computing, there are no limits. So I believe that this is only a small percentage on about how we can use AI in medicine. So I will give you some examples. For example, dermatology. So we know that, for example, there are some moles that and beauty spots that are okay, and some that are dangerous. Okay, so how can we teach AI to distinguish between good moles or moles that may be dangerous with a lot of data? We have to give the AI a lot of pictures, and we have to tell AI, this is a good mole, this is a mole that may be dangerous. And in this way, the AI autonomously how to characterize a good mole or a mole that may be dangerous. For example, we can use AI also in cardiology. We can apply it to the ECG signal, and the AI is able to see from the ECG, maybe if we have a very structured data and a very high quality data, maybe the AI will be able to evaluate from certain features and characteristics of the ECG signal if this person maybe in the future risks to have a cardiac attack. Or maybe we can also, there is a company, for example, in Israel that is called Vocalist that is using AI applied on voice. It means that it knows how to analyze the tone of voice of a person and evaluate if this person has some respiratory problems or cardiac problems. I would say that also the radiology is a huge field where we can apply AI. A lot of companies are now teaching AI to read autonomously the MR and the CT images and say if there may be, for example, a tumor. But I also like to say that the AI is not only to support the doctor, but is also to support the patient. So for example, we can use AI in telehealth, and we will see a small example later. So absolutely, AI is super helpful in medicine, will be even more helpful. We saw only 1% of it.
Thank you, Mr. Misano. I know that you were born in Italy, in Rome, actually, and but you moved to Israel, and now you live and work there. I know that you are a young entrepreneur in the field of health and medicine, and how much and how is Israel focusing on innovation in this sector? Could you provide some examples of advanced Israeli technologies in the field?
Absolutely, Elisa. Thank you for the question. So as we said before, the most important thing about AI is the data. We need to have structured data and high quality data. So for everyone that is interested in this field and in learning more on how Israel is using data, I recommend you this article by Forbes, How Israel Turned Decades of Medical Data into digital health gold. And how did Israel do it? Actually, the cool thing about Israel is that there are HMOs. It means that the vast majority of the population uses HMOs, and these HMOs record all the data. So every time that we have to go to the doctor, to a general doctor, to a specialist, we do an exam, we do an MRI, we do a CT, all this data is stored in structured databases. It means that Israel has access to very, very high quality data that we can use to train AI and make better precision and better predictions in the future. For example, this is just an example of a study published by one of the HMOs called Maccabi that shows that Maccabi managed to train an AI to predict, to see the results, the simple results of blood tests that are all recorded in the HMO database, and predict from these results if the person will have a colon cancer in this case. So I'm going to tell you a little bit about some of the companies that in Israel are pioneers in the field of AI applied to medicine. In particular, for example, Medic Vision is an Israeli startup that developed the IQMR, Intelligent Quick MR. This is an AI algorithm that works on medical images and allows 40% faster MRI scans and 35% increased productivity, while also enhancing image quality. And the cool part about this software is that it is vendor agnostic. It means that we just need to have one of these systems in one hospital, and the system receives the images from all the MRI machines on site. And also, of course, the IQMR is FDA cleared. All right, so how it works? So because we are doing the scan with 40% less time, the images that are gonna come out from the MRI scanner are a little bit noisy. So the IQMR receives the fast scan noisy images from the MRI scanner and automatically processes the images and sends the high quality images to the PACS. In this way, the radiologist receives a precise image and is able to do a precise diagnosis, a precise evaluation. Another very interesting product is called titolo. It's a product that is FDA and CE certified, and it was nominated between the top 100 inventions in 2019 from the Times Magazine. And it is used by a lot of families in Israel, in Italy, in the US, and is designed for non-professionals. So this is a telehealth device that allows to do a televisit from remote. So the patient is at his home and the doctor is in his office. Just by opening his computer, he can connect to the device that is at the home of the patient and can do a remote visit. For example, this device can do a pulmonary and cardiac auscultation, fever monitoring, heart rate, and can provide HD images of the skin, throat, and ear. So let's see, for example, how it works. We will see here an example of simply a child that is not feeling so well. So luckily his mom has the titolo at home. So this is the device, and the mom opens the application. She requests a televisit, a live televisit with a doctor, and now we will see how it works. So these are some of the common conditions that the device can diagnose. There you go. Here we see that the titolo app with the doctor, we can see the doctor live, and we can use the device. Okay, so for example, here the mom puts the device on the chest of the child in order to do a cardiac and pulmonary auscultation. So let's see really a use case. The child is not feeling well. The mom opens the phone, opens the app, requests the live call with the doctor from remote, and the doctor instructs her on how to use the device from remote. Here we see the mom, for example, showing the ear, the throat, and the cardiac auscultation of the patient. And of course, the doctor sees all the data from his platform. Where is AI used in this case? It is used to tell the mom the confirmation that she's taking the right picture of the ear, because the mom, of course, doesn't know. She doesn't know if the images that she's taking is the right one to do an evaluation. So the AI indicates to her when the right part of the ear is shown in the camera. Let's go ahead. And this is another very interesting Israeli technology. Let's see the video. So this is a product that runs exclusively on the smartphone camera. Only on the smartphone camera, no devices. The technology isolates a zone of the face below the eye, and is able to evaluate and see the minimal changes in the color of the skin. And from here, as you can see from the video, it extracts the heart rate, the oxygen saturation, the respiration rate, the HRV, and the stress level. So with this system, no devices are needed, no internet is needed, no data transmission, so the privacy is protected. And it's a technology, of course, AI-based, and it is implementable in one day on millions of phones, because it can be implemented also in other apps. Another interesting, maybe the last innovation that I'm gonna show you is Easy Biotech. Easy Biotech is a startup, half Italian and half Israeli, and I am actually the CEO of this startup. We are developing a kit to do a urine exam at home in 3 minutes. We all know the burden of doing a urine exam. We have to go to the lab, do the line, bring a sample, pay the exam, wait for the results, send the results to the doctor. But we are developing a kit to do everything in 3 minutes at home via a kit that can be bought in the pharmacy, and an app. All right, so let's see how it works. Okay, let's see the video. So simply the patient downloads the application. Okay, the patient is, let's go. Yeah, the patient is instructed. No, sorry. Let's show it again. All right, so let's see the video. So the patient downloads the app. The patient is instructed to urinate in the cup provided in the kit. Then the patient should insert this color board that we see on the right inside the sample. And inside this color board, there are 10 different reagent squares that change color based on the components that are in the urine. All right, so it takes some seconds in order for the color change to happen. So there is a countdown in the app, and then the patient is required from the app just to take a picture of the color board. There you go. The patient takes a picture of the color board, and the app automatically analyzes the color change and is able to give the results of the exam. So why is this important? This is important for to detect diseases like the chronic kidney disease. The chronic kidney disease is divided in 5 stages. And in the first 3 stages, no symptoms happen, but the disease can be diagnosed by a simple urine test. The problem is that people don't do urine tests. So the 70% of the people that are diagnosed with CKD are diagnosed in stage 4 and 5 when it's too late. All right. So with this device, with this kit, we hope to make more people do urine tests, because it is very important and can be and can save it can save a lot of lives. So this was a little glimpse about the how in Israel AI is used for health, to improve the patient's health. Of course, there are many more examples. So if you are interested more about it, just contact me on LinkedIn. Thank you so much again, Elisa, for the interesting questions, and to Bracco Group for the invitation.
Thank you, Mr. Misano, for your very interesting presentation. Okay, so we have come to the end of this meeting. We have explored the new frontiers of medicine, and we have seen how important precision diagnostic is to achieve the goal of personalized medicine, which is to guarantee each patient the most suitable treatment. So let me warmly thank all the speakers for their participation today, but also for their everyday work in their labs or in their companies. And of course, I deeply thank the hosts of Bracco Group who allowed us to take another step together towards the opening of Expo Dubai 2020. Arrivederci.